All right, back again. Here we go. Last type of genetic cross for you guys. So hopefully you're following along. And I know yesterday's notes that you guys copied were probably like, whoa, a lot going on there. Kind of what dihybrid crosses are. So we're going to kind of incorporate everything that you guys know up to this particular point. So with dihybrid crosses, we could do trihybrid crosses, and we could keep going and keep going and keep going as well. But what we're going to do is we're going to follow the inheritance, meaning the passing on of these traits, but we're going to do two traits at the same time. So this is kind of what Mendel did with his pea plants. So remember, Mendel was our Austrian monk. He's the one who's basically our father of genetics, and he studied pea plants. And with this, he looked at traits like your stem shape or your stem length, your pod shape, your seed color, your seed shape. And as he went through and crossed these particular plants, he came up with alleles for each of them. So alleles is something that we've been talking about throughout each one of these particular crosses, but I just want to set it straight for you guys. An allele is a form of the trait. So for instance, if we were talking about height of pea plants, I can say that tall is dominant to short. So my alleles there would be tall or short, my forms of that particular trait. For seed shape, I can say they're wrinkled or they're round. Seed color, they're yellow or they're green. And those are all the different alleles or forms of that particular trait. So as Mendel went through and did this, he started coming up with some laws. And one of the laws is the law of segregation. And with this, he says that paired alleles are going to separate during the formation of gametes. So during that process of meiosis, and they're lining up during anaphase in 1 and 2, and then they're going to go separate out. So we're going to go through and separate not only seed shape and seed color, but then we can also separate out the alleles for each of them. So with shape, we could say that round and wrinkled. So maybe I have a heterozygous. So these particular alleles are going to get separated. They're not going to get both the big R and a little r from that particular pea plant. Remember, one comes from mom, one comes from dad, even in pea plants. So that plant is either going to get a big R or it's going to get a little r. With color, we're going to talk about G's here because we're going to talk about green and yellow. It's either going to get a big G if we're talking about heterozygous, or it's going to get a little g. It's not going to get both of those. The next law talks about independent assortment. So here we're talking about two traits. With this, both these traits are not inherited as a pair, meaning if you get the dominant for seed shape, doesn't mean you're going to get the dominant for seed color. And that's what independent assortment is talking about. We're going to kind of separate them out. And this increases genetic variation. So I might get the dominant for shape, but I might get the recessive for color. There's a whole bunch of different combinations that you guys will see as we go through this particular one. So this is what's really going to increase genetic diversity in all populations, not only plants. So the next page, we kind of go through a little example using those alleles that we just kind of talked about. And we're talking about, with these particular plants, Mendel crossed plants that were homozygous, round yellow peas, with plants that were homozygous, recessive, wrinkled green peas. So a lot going on there, and that's what we kind of want to figure out. There's two ways that you guys should have seen as you're going through the notes that we could do this. One way is where you do a giant 16 box Punnett square. I'm not a big fan of that. I kind of get lost trying to count everything up. The second way deals with a little bit more math, so it depends upon which way you like better. I don't care which way you use. You're going to get the same answer no matter what. Do what works best for you. So the first way we're going to do is with the two different Punnett squares. We have two different traits, so let's kind of separate them out. And to do this, we're going to do something called probability, and we're going to use the law of probability, meaning what's the likelihood that something's going to happen? What's the likelihood that we'll get two of our dominant traits? What's the likelihood we'll get a dominant and recessive? What's the likelihood we'll get a recessive dominant? What's the likelihood we'll get two recessive traits together? That's what we're going to be looking at. And in order to do this, we're going to do a little bit of multiplication. And you guys are going to have an activity with this later in the week as well. So let's say I take a coin, and you guys know on the coin there's two sides to that coin. There's a heads and there's a tail. Let's say I flip that coin 3 times in a row. I want to know, what is the probability that it lands on heads all 3 times? Well, every time you flip that coin, the first time I flip it, I have a 50-50 shot. I have a 1 out of 2 shot of it landing on heads. Well, each event is independent of each other. The second time I flip it, it doesn't matter what happened the first time. I still have a 50-50 shot. Third time I flip it, it's completely independent of what happened the first 2 times. I still have a 50-50 shot. So for that, we need to go through and we need to multiply it. So since I'm flipping it 3 times, I have 50%, 50%, or 1 out of 2 for each of these. I'm going to go through and multiply it. So I have a 1 out of 8 chance of it landing on heads all 3 times. That's what we're going to kind of be doing with these particular Punnett squares. So let's go back to steps that we know how to do. We know our 5 steps. We know how to set up our keys. We know how to set up parental crosses. We know how to do Punnett squares. We know how to do genotypes and phenotypes. Then we're going to kind of add a next little step there. So the first thing we need to do is to set up our keys. So we need to go back and look at the problem, because that problem is going to tell me what is dominant, what is recessive, what are the traits that I'm looking at here. So we're talking about homozygous round yellow peas, and we're talking about wrinkled green peas. So my 2 traits, I have either round versus wrinkled, which is going to be for my shape. So my 2 traits, I have one for shape, they're either round or wrinkled. The second one, I have yellow and I have green peas. So the second one is going to be color. I'm separating them out into 2 separate keys. Now I need to figure out what is dominant and what is recessive. Well, the first one says homozygous round yellow. Homozygous also relates to yellow. So I have homozygous round, homozygous yellow. Doesn't really tell me too much there. But when I look at the next one, it says the plants were homozygous recessive wrinkled. So I know that wrinkled is recessive to round. Round is dominant over wrinkled. It's a regular monohybrid cross. I'm going to use ours. Big R is round. Big R is round. Little r is wrinkled. That's what my cross told me. The second one, for color, this homozygous recessive not only goes with wrinkled, this homozygous recessive also deals with green. So because of that, I know that yellow is dominant to green. I'm still going to use G's. I'm not a big fan of Y's. So I'm going to say big G, which is dominant, is yellow. Big G. My hybrid or heterozygous choice is also yellow. Little g is green. So I have step 1 done. 2 separate keys for those 2 separate traits that we're going to put together in this dihybrid cross. So when we look at the next page, we then need to do our parental cross. I'm going to kind of stack this here together. Hopefully it comes up a little bit. Okay. So going back and looking at my parents, I'm going to do a giant parental cross here. So I'm going to say, okay, well, here's plant 1 for both shape and color. Here's plant 2 for both shape and color. So if we go back to that cross that you guys had on the top of the previous page, it says parent number 1 was homozygous round yellow. Homozygous round, big R, homozygous yellow, big G. I'm going to put both of those together. We're looking at both traits together here. So big R, big G. Plant number 2, homozygous recessive wrinkled green peas. Homozygous recessive, little r, green, little g. That was plant number 2. So now it's like, whoa, we got a lot of these alleles here. We gotta want to separate them out. And that's what I want to do. I want to make it something that I know how to do. At this point, I don't want to mix all this stuff together right now. So I want to say, okay, well, I have parent number 1 that has a trait for shape. Parent number 2 has a trait for shape. Let's put those 2 together. Let's stick my R's together here. So here is my shape gene. Here is my color gene. Parent number 1, big G. Parent number 2 is little g. When I get to that point, I'm like, oh, I know how to do this. I've done this before. Here's my regular monohybrid cross. I'm just going to do it twice with 2 different traits at that particular point. So as we're looking at it, I say, oh, well, big R cross with little r. There's my parents, parent 1, parent 2. Go through. Remember, independent assortment. Just because I get one of particular dominant trait doesn't mean I have to get dominant of the other. I'm separating them out. It increases genetic diversity. So as we go through, we do the Punnett square. The steps are still the same. Here's my Punnett square, genotype, and phenotype. So here's my advice. When you get down to that phenotype, put it in fractions. So I have, as I go through, big R, little r, all of these F1s that I see here, all those F1s are going to be big R, little r. So genotypically, I have 0, 4 to 0. Phenotypically, how many are round out of those 4? Well, all 4 out of 4 are round. How many are wrinkled? 0. I don't have anything in there that is wrinkled. So 0 out of 4. I do the same thing with color. Big G, little g, put it in that Punnett square. Genotype, phenotype, I put the phenotype in fractions. How many are yellow? All 4 out of 4, because once again, these are all those F1 offsprings. So looking at it, I can say, oh, well, all those babies are going to be round and yellow. That's the only alleles or genes that we have in that particular plant there. But we're going to use probability in order to figure it out, because we can have different combinations here, because this is where my probability comes in. I want to see how many plants can be round and yellow, both traits at the same time. How many can be round and yellow? There's my two dominant choices. I have other choices. How many can be round and green? Dominant, recessive. Well, maybe I have some plants that are wrinkled and yellow. There's my recessive choice and my dominant for color. How many can be wrinkled and green? My two recessive choices. So whenever I do this, I have 4 different combinations that I can create here. Now we know this one's kind of easy because I know what all my plants are going to be, but we're going to use it as an example so we can go through and create our next F2. So we're going to find the probability of both traits at the same time. And that's going back to that coin flip that we have. So I usually will go through and kind of write it out. And I'll say, okay, well, how many can have both dominant traits at the same time? How many can be round? How many can be wrinkled? Go back to your phenotypic ratio where we have them in fractions. I said, oh, round, I have 4 out of 4. Yellow, I have 4 out of 4. Multiply them through. That means that I have all 16 out of 16 that are going to show both those traits at the same time. How many of them are round and green? So round is 4 out of 4. Green is 0 out of 4. Anything times 0 is still going to be 0. None of my plants are going to show both of those traits at the same time. Same thing with wrinkled and yellow. Wrinkled is 0 out of 4. Yellow at 4 out of 4. Still 0 out of 16. Wrinkled and green, I have 0 out of 4 and 0 out of 4. Still gives me 0 out of 16. So what I end up doing is I basically will go through and I add them up and I make sure that I have a total of 16 out of 16. So that's saying that all of my F1 offspring have to be round and yellow. It's the only thing I possibly have. So let's go to the next step. It says, okay, well, now let's go through and cross 2 of the F1s. Cross 2 of my F1s. There's my F1s. If these were all different, you would just go through and pick any 2, but they're all the same. So as I'm going through, I'm going to say, okay, well, I'm going to take this one and take these 2 alleles and cross it with these 2. And what that means is that my F2 generation that I'm going to create is coming from my parents. My parents are those F1 babies. I'm using the same case. I'm not going to rewrite that for you guys. Round versus wrinkled, and then I have green and yellow there. Yellow is dominant to green, remember. So my F1s, I have big R, big G, little g. Big R, big G, little g. So there's parent 1, there's plant 2 or parent 2. Let's do the same thing. Let's separate out our traits. So I'm going to separate out for shape. I'm going to separate out for color. So once again, parent number 1 for my shape is going to go right here. I'm going to take parent number 2, separate it out for shape. I want to keep my letters the same. So let's keep my R letters together or R genes together. For my color, parent number 1, big G, little g. Parent number 2, big G, little g. Still the same. Keep them together. Now I'm creating that F2 generation. I know how to do from this particular step. Punnett square, genotype, phenotype. Keep that phenotype in fractions. So as I go through, big R, big R, big R, little r, big R, little r, little r, little r. I end up genotypically with a 1 to 2 to 1 ratio. Easy first type of problem that we did. Phenotypically, big R, big R, big R, little r is round. Keep them in fractions. So I have 3 out of my 4 possible F2s that are round. 1 out of 4 that's going to be wrinkled. If I look at color, same thing, 1 to 2 to 1, which gives me 3 out of 4 that are yellow. 1 out of 4 that is going to be green there. Now we need to calculate the probability. This just says I can't stop there. I'm just saying, okay, 3 out of my 4 plants are round, 3 out of my 4 plants are yellow, but how many are going to show both traits at the same time, which is what we want to look at here. I can't just leave them at that particular step. So now we got to go back to that probability again. And when we combine that probability, how many can be? And you'll always have, if we're doing a dihybrid cross, 4 possible combinations. So we have ones that can be round yellow, round green, wrinkled yellow, wrinkled green. And I usually will line them up like this so I can kind of see the combinations there. When I write them out, how many are round? You already have it written down in your probability, 3 out of 4. How many are yellow? Still 3 out of 4. So how many can be yellow and round at the same time? 9 out of my 16 will show both of those traits, probability-wise, will show both of those traits at the same time. How many are going to show the dominant recessive trait, round, green? 3 out of 4 for round, 1 out of 4 for green, gives me 3 out of 16. How many are going to be wrinkled and yellow? Recessive, dominant. 1 out of 4 for wrinkled, 3 out of 4 for yellow, still gives me 3 out of 16. My 2 recessive choices, I've got wrinkled, 1 out of 4, green, 1 out of 4, 1 out of 16 for that particular 2 recessive choices. This is where I say add it up. Make sure that your numbers equal 16 out of 16, that your math is not a little wacky there. What you guys are going to see is that you will always end up with a 9 to 3 to 3 to 1 ratio when I'm crossing parents who are hybrid for both traits. That's what we did. Big R, big G, little g. Both parents were that. Anytime you guys have a Punnett square or dihybrid cross and you're looking for that ratio there, it will always be a 9 to 3 to 3 to 1 ratio. Why is that important? Because if you're doing it on a test and it's multiple choice test, and I say, okay, cross these 2 parents, blah, 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 and I ask you questions about it, you're like, oh, I already know what the ratio is going to be. I can answer those questions without doing the problem. If I give you practice problems, I still want to see the work. If I'm asking you to turn in the work, I need to see the steps that go with it. Don't just put 9 to 3 to 3 to 1 on it. So I know that I have a 9 to 3 to 3 to 1 ratio for dominant, 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 recessive, recessive, dominant, recessive, recessive. How can I ask questions on this? Well, instead of doing 16 pea plants, maybe Mendel had 128 offspring. Of those 128 offspring, how many of them are going to have the particular traits of wrinkled and yellow? So you go, how do I do that? Wrinkled and yellow. I know I have 1 out of 16, but now I have 128 offspring instead of my 16 offspring. So what I want to do is that I kind of want to go through and figure out, well, if I have 16 offspring, how many times can 16 go into 128? So let's take 128 and divide it by 16. And I know that basically is going to give me 8. So I'm doing this kind of 8 times. So if I'm looking for wrinkled and yellow, which is this number right here, I'm going to times it by 8 instead of just the regular 1 section of it. I'm making 8 times the amount of it. So if I'm doing that and I have 3 out of 16, well, how many would I have at 8? I'm going to have 24 offspring because I'm going to do 3 times 8, and that's going to give you 24, not your 28 there, 24 as your possible number of offspring that can show both those traits at the same time. Okay, usually if I give you a problem like that, I'm going to give you something that is divided by 16. I'm not going to make you guys do fractions or anything like that. So maybe I'll do 64 or 32 or something along those lines. There is a second way to do this. And usually if you look online, this is kind of the way that they always show you. I am not a huge fan of this way because I kind of get lost writing letters in boxes and I don't like the giant Punnett squares. And it gets a little hard to count for me at the end. You are more than welcome to do this. This also is another little math method. So this is a FOIL method. So same thing, I'm going to take my F2 generation. We have big R, little r crossed with big G, little g, plant 1. Big R, little r, big G, little g. Remember that if when we do our Punnett square, I need to do a giant Punnett square with 16 boxes. One parent goes down the side, one parent goes across the top. It still does not matter who goes where. We are using what is known as the FOIL method. You might have heard it in math. First, outer, inner, last. So just looking at one parent, one plant here, I'm going to try to combine these particular alleles together. So when I break them down, I know that this particular plant is either going to give a big R or a little r. It's either going to give a big G or a little g. I got to see those combinations. So first, I did it in red here. Big R goes with big G, the first of the second set of traits. So big R, big G. Outer, big R, little g, the outermost letter there. Well, maybe it's the little r gene or allele that gets passed through. So little r, inner, maybe goes with the big G. Or maybe that last little r goes with little g. So first, outer, inner, last there. And that's what you guys are combining there. And I'll do the same thing across the top. Now we have to go through and combine them all together. Stick your capital letters first, but keep the traits together. So I will always sticking my R genes together and my G genes together. My R's will go before the G's, capital letters before the lowercase letters. So big R, big G, big G. Big R, big G, little g. And you would keep going and you would fill in all your boxes. Then you need to add up your phenotypes. So I would usually write them all out. Okay, how many could be round and yellow? 2 dominant choices, round and green, dominant recessive, recessive dominant, recessive recessive. And I would go through and count them. When you count them, put a little check mark or an X in the box, knowing that you already counted that particular box. So I would go, oh, big R, big G, that's round and yellow. 1. Big R, big G, little g, oh, it's still round and yellow. And I would go all the way through. What you guys are going to see is you're going to end up with the same ratio that we just did with the 2 Punnett square method there. So like I said, you guys are more than welcome to do whatever one that you like. But for the 2 that I'm going to give you guys, I do want to see the work that goes along with it. And then we'll probably do one that deals with blood types a little bit more tomorrow. So the second attachment that you guys are going to see is for the worksheet that you have 2 practice problems on. It's going to look like this. I'm only asking you guys for 2, but I do want to see the steps. Once again, I don't care if you use the Punnett square method or you use the probability method. Okay, and then we'll meet later today with a Google Meet. If you guys have questions, please come see me and we'll go through anything that you have. Remember, that test is coming up shortly.